Alright guys, yesterday I threw a lot of information to you about how DNA and RNA are the same and how they're different. So this screen just reviews what we did yesterday. Remember, double-strand, single-stranded, different sugar. T is different, as in RNA it is a U. The phosphate's the same. The fact that they are nucleotides is the same. And the RNA can act as the messenger and head to the nucleus. To the ribosome. Okay? So then, we covered this yesterday, so actually turning DNA into a real living live trait is called protein synthesis. And that takes two parts, transcription, which we barely started yesterday, okay, and translation. So I'm going to go through those parts in a little bit more detail today, okay? So let's start with the messenger RNA first, okay? This is turning DNA into RNA. Honestly, all you have to remember is obviously the correct term, transcription, and think about what those word parts mean. Trans means to change, and scription means to write. So we're going to change the writing. So all you have to memorize for this that is different is remember that A pairs with U. Okay, so let's practice one of those. And you know what? I messed this up here. Let me change this. This needs to be like a T, right? Because there's no RNA in, no T's in DNA. Okay, so let's leave it like that. All right, so from what we've done before, if I asked you to write the other side of DNA, okay, let's start with that. T pairs with what? You know that's an A, okay? I'm going to skip this one for a minute. How about the C? Pairs with G, right? Okay, come on. The G pairs with the C and another C. Okay, this T pairs with an A, and the C pairs with a G. So the only difference when you're making RNA is the fact that when you see an A, it's now not going to pair with a T. It's going to pair with the T's cousin, which is a U. Okay? So you should have U's here, like this. All right? So what we have right here, okay, this strand right here is messenger RNA. It's a single strand, and it's going to carry the instructions for whatever DNA wants done at this particular time. Okay? It is way smaller than the whole DNA strand. Unlike with replication, you're not copying the whole thing. You're only copying particular genes. So as an example, let's say you did like I did when I was in 8th grade and I was riding my bike through the water where my dad was washing his car, and he's like, Christine, you can't do that, you're going to fall. So indeed, I did slip and I landed on my ankle and I broke it in a couple places. Okay, so obviously my DNA went to town making new bone cells, new muscle cells, right? All kinds of things to repair that damage. So we didn't have to copy the whole DNA strand. We only needed the particular portions of the DNA strand that I needed to heal those particular parts. Okay? So transcription is much faster. It is dealing with much less data, okay, in terms of genetics. And then remember the A's pairs with U's instead of T's. Okay? So it's that simple. All right? So let me show you this part here and give you a couple more vocabulary words. So this is meant to represent our nucleus here, okay, this purple wiffle ball looking thing. And this single-stranded thing here is RNA, okay? How do you know it's RNA other than I just said it and it's typed out? Well, you have a couple clues. One, it's a single strand. It can fit through those little membrane holes and get out. Secondly, you see U's up here instead of T's, okay? So those are two clues that this is RNA. Now, in a little while, we're going to learn about something called a codon, and codons we're going to need to turn, to tell what the ribosome which particular amino acid to make, okay? So I want you to think of codon like a code. You know how we have 3 numbers to open up our lockers here at school, okay? This is very similar. Every set of 3 nitrogen bases is called a codon, and we actually have 64 different possibilities for codons, and I'll show you that in just a minute. And then those codons in turn end up being a code for a particular amino acid. Okay, kind of think of it like when you go to the grocery store and you scan your green beans and your chicken wings and your package of French fries, okay? That particular barcode tells the register which item you've purchased and how much it costs and whether it's on sale or not, okay? So think of these little codons as specific identifiers for the 20 amino acids, and I'll show you all that in a minute, okay? So this messenger RNA can now head over to the ribosome, which is represented by this blue blob here, okay? Then the ribosome is going to read those codons and say, oh, okay, DNA needs a threonine, or DNA needs an alanine, so I'm going to bring that to it, okay? So remember our types of RNA: messenger RNA, tRNA, it actually looks like a T to me, coat hanger, and then the rRNA is the ribosome itself, okay? And then if you'll notice, on the tRNA is something called an anticodon. I want you to think of it as opposite, okay? So as you can tell here, our codons on RNA were AUG, UUC, and the next one coming up is AAA. Well, if you look on the tRNA molecules, the anticodons are the opposite of those, kind of like how we did complementary strands on DNA. So the opposite of AUG would be UAC. The opposite of UUC would be AAG. And then you can tell this UUU is getting ready to pair up with this AAA. All right? So those codons meet the anticodons. And the only purpose of the anticodon is it tells the tRNA, hey, I'm in the right spot. This is where this amino acid lysine needs to go, in this particular spot. Because remember, as we talked about before, the order of your nitrogen bases is what gives you your unique characteristics. So obviously the order of the amino acids would mean the same thing. So if this was placed here instead of here, it would be a huge mistake, as we call a mutation, which we'll do later on. Okay? All right. So translation is the process of turning RNA into a protein. So far we've done transcription, which is turning the DNA into the RNA. And then the next step, the RNA leaves the nucleus and heads over to the ribosome, okay? And then the ribosome will turn that RNA into a particular protein, which gives us our traits. Okay, so that's the process that's going on here. And then this chain-looking thing here is basically, it's called a polypeptide, and all that means it is a protein. Okay, and proteins are held together by peptide bonds, not hydrogen bonds like DNA. Peptide bonds are extremely strong. If you guys have ever tried to eat a piece of meat that's very tough and hard to chew, you know what a peptide bond is, okay? Think of like beef jerky or something like that. That's really tough because it's proteins, all right? Okay, so guys, here's a picture of the entire thing, and I know this is going to look difficult to you, but let's bear with me, okay? So here's the nuclear membrane. So I want you to think step one is all in the nucleus, okay? Let's do some things that I know you know. Okay, the red double-stranded thing, you know that as DNA, right? Okay, this is an enzyme. You don't need to know the name of it. But what it is creating is this blue strand. Notice it's single and it's a different color, which is meant to represent the different sugar. That's your messenger RNA, okay? So making DNA into RNA is one of our trans words. That's transcription, all right? So now the RNA can leave the nucleus and go to part 2 of protein synthesis, which is translation. We're going to change the language. We're going to change the A's and U's and C's and G's to amino acids, lysine, tryptophan, glutamic acid, and so on, okay? So here's our messenger RNA strand. Remember, every 3 pieces is called a codon, okay? This crazy looking thing here is our tRNA molecule, and it meets up at the ribosome, because remember, the ribosome is what makes the protein, okay? This pink chain here is your developing protein, okay? The tRNA carries the anticodon, which is the opposite of the codon, only so it knows it's in the correct spot, and that is translation, okay? So transcription, turning DNA into RNA, translation, turning the RNA into an actual protein, okay? So, guys, one of the things you're going to have to be able to do is to turn the A's and U's and C's and G's into an amino acid, okay? Now, this looks a lot more complicated than it is, okay? So I want to put one down here. Let's say we're going to find out what AUG is, okay? Now, it doesn't matter what chart they give you. They're all going to be similar. They may not be arranged exactly the same, but they're always going to be the exact same answer no matter where they are in the chart, okay? So notice you have the first base here, your second base here, and your third base here, okay? So what's our first nitrogen base in our codon? It's an A, right? Okay, so here's how I want you to look at this, and I'm going to make this really big for a minute. Is that big enough? Yes, okay. So what the A tells me is that my answer of the amino acid I'm looking for is somewhere in this row. So I have just narrowed it down from 64 possibilities to 16. That's pretty darn good. Okay, now what is the second base in our codon? It's a U, correct? Okay, so the U tells me what column it's in. See, the second one is the column. So I'm going to highlight this. Okay, now what that has done for me is helped me out tremendously. Now I've gone from 64 to 16 to 4 because my answer is somewhere in here where these two overlap. Okay, so that's what the third the third nitrogen base is for. So if I look down, the G is the second one in the box. So the second one in the box is methionine. Okay, so that tells me that AUG is the amino acid methionine. Okay, this one is also called START, and I need you to memorize that for me. Okay, and an easy way to do that is think about when we normally start school. It's in August. Okay, so AUG is always a START. Okay, because think about it this way. If you're working on an assembly line, the assembly line has to be turned on. So think of AUG as the start codon. Okay, and you can even put that next to that if you'd like. All right, so see, it doesn't matter how the codon charts look. Let's try AUG again. Here it is. Okay, and it tells you it's methionine. MET is just the abbreviation for that. All right. So the ones that are on your EOC are actually very easy because the the codon is always right next to it. Okay. All right. So if you guys want to practice some of these, it would probably be best for you to either print one of your messenger RNA codon dictionaries or always have one pulled up. Okay, because this is a very important part of that. All right, so I want you to give some of these a try when you have a chance. I'm not going to waste the minute and a half I have left on those, but we'll get some more practice with that, okay? All right, we've done all these. Okay, so I do want to actually finish on this particular screen with you, okay? So see how it says turn these DNA strands into RNA strands. So guys, it is telling you to actually write a U instead of a T, okay? So just remember, T pairs with A pairs with U, G, 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 C, and then like 5 U's in a row, and then a T. Okay? Same thing here. Same thing here. All right? So then, if I got you to go further and actually figure out what amino acids is made, what you would do is divide it into 3s, right? Remember, our codons work in groups of 3. You would do that for all of them. Okay, then you would look up these in that amino acid chart. So we will practice some of that in the Google form, okay, to get really good with that. All right, so guys, here's an overview of that, and this is what I'm going to leave you with since I'm running out of time here, okay? And we will do much more practice with this on Monday and maybe even Tuesday, depending on how well y'all are doing, okay? But it's going to be very important that you play this as much as you need to, you look at the notes as much as you need to, and look at supplemental videos if you need to. I'm sure you guys all have your favorite go-to people to look for. Okay? So give those a try. Give me a call if you have any questions. Thank you.